Right. Okay, so the goal here is twofold. There's sort of the MKDA part and the Neurosynth part. The MKDA part is really sort of more generally the goal of meta-analysis, which is to make sense of the fMRI literature and kind of try to derive some consensus sense of what's going on from potentially sort of fragmentary individual studies. When it comes to Neurosynth, the goal is, sort of the way I think about it at least, is to do this in an accessible, easy-to-use way. So as you'll see, there's sort of, and it's already come up, there's a difference between sort of the Neurosynth philosophy and the brain map / Ale philosophy. And the Neurosynth philosophy is basically like, let's not worry about quality and let's just make it really easy, right? So there's a trade-off there, but that's the basic dimension, I think, on which these differ. There's a meta-analysis, right? I mean, that is one way of doing meta-analysis. You just throw all the activations from a bunch of studies, I think this is emotion studies, at a brain, and you look at that, and of course, it's very easy for you all to draw some sensible interpretation of that, right? It's very obvious, falls off the page. No, that's just, I mean, that's not helpful. If you do some statistical magic via MKDA or some other method, you can generate something like the bottom plot, which is now much more sensible and easier to understand. This is from a paper from Tor Wager's group about a decade ago now, almost. It's been a long time, I guess. And this is a meta-analysis of emotion, and you probably, if you know anything about emotion, you'll look at this and say, like, yes, that makes perfect sense to me. Part of the reason it may make perfect sense to you is because of maps like this that have now been around for 10 years. But the point, of course, is that we want to do this in a quantitative way because the top does not make much sense and the bottom does. And so the tools are really just designed to give us some sort of quantitative ability to distill what's going on. So Tom already talked a bit about MKDA, so I'll just breeze through this. But the basic idea here is you take activations in the literature. So here, every study, let's say in this in the sort of leftmost panel, is every point is an activation. And you multiply that by a hard sphere just to smooth the data. And then you can average over, let's say you have 100 studies. You just take the average intensity at every voxel, which is the proportion of studies that activate for that contrast. And then you can do some significant, or you can compute significance via permutation tests. And I'll walk through some of the details here. In the case of multi-level kernel density estimation, again, so let's say we have 437 studies in this case. Each map has, so the leftmost map might have a whole bunch of activations, and then maybe one study only reports bilateral amygdala and so on. And we want to combine these in some way. So again, we just multiply these by a hard sphere, and then we can average them. So we literally just take the average at every voxel, 1 versus 0 for every activation, right? And so you end up with a continuous density map. And that tells you, for example, well, in the amygdala in this case, maybe 12% of studies activate, right, for this contrast of emotion versus some baseline. And you can see just by looking at that already that there's some structure there. So you can see, well, it looks like the amygdala is more activated in studies of emotion. But then what we can do is we can randomly shuffle the blobs around. So we create a null distribution. And depending on the correction procedure, you might say, take the maximum intensity value at the 95th percentile over all these replicates. And so you're basically referring the observed values to some null distribution. And if the observed result passes, or if it's greater than, let's say, some particular threshold in the null distribution, then you would say you have a significant effect, right? And so you can compute p-values in this way and end up with a map that looks like this. And that's the map you would probably report and say, well, now we have something that we feel comfortable is maybe quote-unquote real. MKDA allows you to do study weighting. This has become much less important in recent years because almost everything now is, at least methodologically, I think there's much better quality control in terms of what gets into the literature. But at least historically, the idea was initially that random effect studies used to get a larger weighting because in the early days of fMRI, people were doing fixed effects analyses, and so the idea was just to downweight fixed effect studies. The general principle here is really that you can adjust for study quality, right? And you don't have to worry, you can, any way you can quantify differences in quality, you can adjust for, and you should. If you think there's some reason that some studies are just not trustworthy or should be trusted less, and sample size is one dimension that Tom already talked about as mattering, right? Then you can quantify that, and so you have the ability to do that. Typically, this is not weighted by peak z-score, and Tom already alluded to this, right? I mean, one problem is that we don't get continuous maps, and so these analyses are not generally based, and this is true for either of the methods you heard about, are not based on continuous effect sizes, and that's, as Tom said already, that is an issue. but that's something we live with. In this case, we are just essentially binarizing the data and saying there's at least one activation near this reported coordinate, which is not optimal. And I think you'll hear a bit later about methods. Image-based meta-analysis would be better, as I think everyone here in this session is going to tell you over and over again. So ideally, we would use continuous maps, but we don't. Similarly, Neurosynth, you can't do that right now, and not really any other method, as far as I know. No database has a very large set of these images. I already mentioned this. Thresholding is done via Monte Carlo procedure. And in general, if you're looking to using MKDA, there are free downloadable tools. If you work primarily in MATLAB, there are free tools from Torwager's lab. There's some documentation on their wiki. There's a mailing list, which I would encourage you to use. And there's lots of features of the implementation that I haven't discussed here. So it's nicely integrated with some visualization and reporting tools. It implements other statistical approaches too, and it does give you some tools for editing and study processing. So you already heard about this a little earlier, and you have tools for sort of editing the studies that are going to go into your meta-analysis. So that's MKDA. Let me talk a little bit about Neurosynth, which is something I'm more familiar with and have developed in recent years. So Neurosynth is first and foremost a website, which many of you may have seen already. You can go to neurosynth.org. And it's really intended just as a platform for doing meta-analysis quickly and on a large scale. And Angie already covered this, and so did Simon in a little bit. So you can think of Neurosynth as a website. You can also think of it as a database. So it's a database of 11,000 or more than 11,000 studies and 400,000 activations. So it's very, very large. As Angie mentioned, the quality is rather poor, but if you want quantity over quality, then you have it there. And it's also a set of, there's underlying tools written in Python for simple filtering and meta-analysis operations that you can also use in theory to do new meta-analysis, although I would not actually recommend this. This is more sort of, we use these tools to generate all the maps that are on the website, so we make them available. But if you're doing your own meta-analysis, I would not suggest you start here. It's also a Twitter account, which is very important. So when Neurosynth goes down, sometimes it's kind enough to notify you. And then you don't have to waste your time, try to refresh frantically until something comes up. So how does it work? I'll walk through the basics, and you'll see it's actually very, very simple, at least sort of schematically. What we do is we follow it, and if it looks like a duck in Quack's philosophy, right? So if it looks like a duck and quacks like a duck, then it is a duck, by which I mean anything that looks like brain activation is brain activation, we say. So this is a kind of an unusual and very, frankly, it's a bit strange assumption because it doesn't seem like it should work. If you think about what goes into producing a published article, right, it generally is not the case that you could just pick off like XYZ coordinates and understand them. Right? You don't know whether the population is, whether it's a positive activation, negative activation. You don't even know if it's functional connectivity, group level studies, whatever. So we have a parser that literally just looks for XYZ-like numbers in sequence within HTML tables of published articles. Right? So that's the basic assumption. But if you're comfortable with that assumption, it makes it very easy to acquire a lot of data very quickly because, for example, here's a typical table. Many of you will have published these kind of things. And you can see that there are XYZ coordinates there, right? So, right here. And this is all we look for, is just these columns, and we pull them out of every article we find. And great, now we have a bunch of coordinates pulled from lots and lots of articles. On the other side of things, we can ask, well, how do you tie this to like function, right? So great, you have sort of a bag of coordinates. Now what? How do you know what this means? We make a similarly simple assumption. It's called a bag of words assumption. So these are a class of methods used very widely in computational linguistics. And we just treat every study abstract. Initially it was full text, but it turns out it doesn't even really matter. So we use the abstract of articles as a bag of words, an unordered list. And so we just say if a term occurs with some minimum frequency, which happens to be once or more in the abstract, then the study is about that, right? The abstract says pain study. It says emotions, an emotion study, et cetera. Non-exclusive, so a study can be about many things. So as I just said, so we show that painful stimulation lists colorful language use, then now this would go into meta-analysis of the words painful, stimulation, colorful language, et cetera. So if you buy those two assumptions, and you don't have to, but those are the assumptions we make, then you're now in a position to do the following. You can now say, let's take the word pain, let's say we're interested in pain, and we will scan the literature for all the studies that use the word pain in the abstract, and maybe there's, I don't know, 500 of those, and then we use our parser to extract all the coordinates. And at that point, you're back in sort of standard MKDA or ALE land, right? You can use whatever tool you want at this point to take that table of coordinates and meta-analyze it. Nothing has changed except the quality is poor, but you have more data, right? So think of Neurosynth as sort of independent of the statistical method used to actually do the meta-analysis. Really, it's sort of a platform that facilitates meta-analysis. But whatever you want to use, you can, and you hopefully will end up with something that looks sensible like this. And this is actually, or was at some point a few years ago, the map you get for pain if you do a Neurosynth meta-analysis. So I'll talk a little bit about what you can do with this. It is a simple, but it's, I think, a powerful framework. So some examples. You may have heard the term reverse inference thrown around. I will sort of talk about reverse inference. Won't get into details. We can estimate the probability of some term occurring in an article conditional on some pattern of activation, right? And so this is typically what you might care about when you get to the discussion section. You say, well, I saw activation in DLPFC. What does that mean? What you're implicitly saying is, what is the probability that you would see some mental state or some process present in your subjects given some pattern of activity? Well, we don't have subjects because this, again, is purely pulled from text, but the hope is that this is a reasonable proxy so that when you see some pattern of activation that suggests there's a high probability that the term language would be used in the abstract, then you hope that's a good proxy for actual language processing in people's heads in scanners. That's an assumption that we don't test directly, but that's the underlying assumption. But it means that we can quantify that, and that's something that's actually fairly hard to quantify unless you have a large set of studies. Because in the typical case, if you only have, say, a bunch of working memory studies, and you never looked at language or pain or emotion, then all you can estimate is what brain regions are associated with working memory, right? And so you can estimate what's consistently associated with working memory, but you do not have the capacity to say what is sort of preferentially associated with working memory as compared to other types of things. And when you have a large data set that has all kinds of things represented, you can do that to some extent at least. You can say, well, let's take all the working memory studies and all the studies of other stuff. You could be more specific. You could say all the studies that are not of working memory and just contrast them and get an estimate of the relative specificity of activation. So you can ask, how likely is a study to use a term if it reports activation in a particular voxel? And so we can of course, also ask, statistically speaking, is there an association? If you just imagine 2 by 2, active or not active at a particular voxel, uses the term, doesn't use the term, well, you can just compute a z-score or a p-value. And so I call this MKDA light because there's actually, at least as far as the website goes, because we're doing this for thousands of terms, there's no permutation, there's no weighting, there's actually no family-wise error correction. We use FDR. You'll get, ballpark the same result, but if you really want to, be confident, then you probably should take extra steps and go beyond this. But I think given the scale of the database, this actually, I think, works pretty well. And so what we call the reverse inference maps on Neurosynth they should be actually called association tests. They're actually, they're just telling you whether there's a statistical association, a non-zero association between the term you see and the brain regions you see, right? So it's a bit of a misnomer, but it is what it is. And this can be fully automated and done for thousands of terms. And this really is the point of Neurosynth, right? I mean, nothing about the meta-analysis machinery is new at all. You've already seen this all, and this has been around for a long time. The idea is really just scale. We can scale this up very rapidly so that we can now do this. We can take any term that shows up in abstract and almost immediately, within seconds, generate a map like this. And if you work in any of these domains, probably you'll look at these maps and say, yeah, that basically looks reasonable. And you don't have to take my word for it. There have been several studies now that have been directly compared sort of manual, very carefully done meta-analyses with the Neurosynth result. Here's a paper from Joe Cable's group a few years ago for both positive reinforcement and negative reinforcement, and they look almost identical. Now, I'm not suggesting that you can replace hundreds and hundreds of hours of effort by pushing a button. This only works when you're talking about very broad things that can be captured by individual terms in abstract. So pain, sure. Emotion, sure. If you want to do a meta-analysis of like mechanical pain versus thermal pain, Neurosynth is not going to help you, right? At that level of specificity, we just do not have enough studies. We would need hundreds of studies for that comparison to work, and we don't have that. So, so you can't replace, you can't use this as a tool to just automatically do meaningful meta-analyses, or at least more specific meta-analyses. But for terms that occur very, very commonly, you do get what is essentially the same representation that you would get using other approaches. Now on the website, we have a bunch of other tools. So it's not just the data we've generated. So we have functional connectivity maps that come from the Brain Superstruct project. And so if you navigate Neurosynth, you can click on any point of the brain and see a functional connectivity map that comes from a large data set provided by Randy Buckner and Thomas Yeo. You can also see the functional coactivation map from Neurosynth, and they generally will look very similar. You can overlay them. You can also ask what's going on in a particular region. So there's this "What's Here" button. If you click that, what that will do is take you to a table that looks like this. So for that particular voxel, it will pull up a table of associations. And this is an unfiltered list, so you can see that most of these terms are anatomical, not surprisingly. But this is telling you that the top associates, statistically speaking, for this particular point of the brain are, in this case, medial, theoretical reasoning, MPFC. And so this is nice because it gives you some quantitative sense of what the sort of top hits for this region are. You will also note if you do this generally that you don't get very specific results. So it's not like you're getting the answer, yes, this medial PFC does theory of mind. You get sort of family resemblance of these terms. And you can say, look, it looks like there's some association there with these terms more so than other things maybe. Okay, so one way to think about what this buys you is you can replace sort of really crummy qualitative interpretation with slightly less crummy quantitative interpretation, which turns out to be important, right? Because I think one of the most useful things about this is it prevents you from saying things that are wildly wrong. It's not super helpful in making positive claims, but at least can tell you when something you're saying is maybe not supported by the data, at least as it exists right now. We provide decoding tools. They're sort of lightweight decoding tools, I would say, but they help for interpretation. Any map you upload to NeuroVault, which I'll show you in a moment, it is automatically integrated with Neurosynth. You can sort of decode it by correlating the map you just uploaded with every meta-analysis map in Neurosynth, and that can give you some sense of what is this most similar to. NeuroVault's already mentioned a couple times. It's a website developed by Chris Gorgolevsky, and I would encourage everyone again to put your maps in NeuroVault, because if you do that, ultimately we'll be able to move away from coordinate-based meta-analysis towards image-based meta-analysis, which is where I think we all want to get to. And so here's a map someone uploaded represented on NeuroVault, and you will notice that there is this analysis button, and there's several options there now. Only one of them is Neurosynth, but if you click it, it will take you to Neurosynth, and then you can see the top sort of correlates in terms of the meta-analysis maps in Neurosynth, and that can help, I think, interpret what's going on. And I find this more helpful because it's based on the whole brain pattern instead of just picking out a region and saying, what does this region do? What does that region do, right? Then you still have the integration problem, like how do you put this together mentally? You can do other things with this. We tried to, the data is all freely available. And so you can take this, you can not care at all about everything I showed you so far and just take this entire database and do things like parcellation, right? So you can ask, well, let's take this entire data set and parcellate it. And here's some nice parcellations we've come up with based on this data set. You can use it as a tool for exploration. So we have some work integrating this with the gene expression data from the Allen Brain Atlas, although I would treat this with a high degree of skepticism. But there are tools for this, and also integrated in NeuroVault, so you can identify gene expression maps that are most similar to the activation maps you just uploaded. Be skeptical. That's all I'll say about that. The key point about all this is that everything is open. So all the extracted data, all the code, all the images, everything is publicly available. The code is available on GitHub. The data set also is. This is all released under a super permissive license or just as public data. You do not need to ask anyone for any permission. You can do whatever you want with it. And again, I think this is a differentiating feature to some degree between Neurosynth and BrainMap. You can take this and run with it and do whatever you want with it. There are no conditions on use. I mean, I like to know what people are doing, so it's, I don't mind if you send me an email saying, hey, here's a cool thing we did with it. But I'm not going to, I won't have any restrictions otherwise. And we do have a mailing list that you can ask questions on. So I will stop there. Thanks.
Well, thanks again. Congrats for also staying on time. It's a little bit of a weird session now. Are there questions, comments?
So I'm not. I like low-hanging fruit, and that's not low-hanging fruit. I think Angie's doing some work on that, and there's other people working on that question. And I think that the right direction to go, and again, I think is what Angie's pushing towards, is using machine learning to actually try and predict human labels. Ultimately, I mean, I agree that we're not going to do better than human labels, at least not in the foreseeable future. And I think that is the right way to go, but I just haven't really invested any effort in that.